Genetically Modified Crops and the World Around Us by Amar Dillon, Ryan Smith, Andy Martisic, and Matt Whitman. Our research question is, how do genetically modified crops affect us as well as the environment?
So the main problem we came across while looking at this research question was that genetically modified foods are destroying the biodiversity in the world, thus leaving it more susceptible to ecological disaster. Firstly, let's talk about what is biodiversity. Biodiversity, as defined by the American Museum of Natural History, is the variety of life on Earth, everything from the genes in organisms to the organisms' ecosystems that they live in. Biodiversity is important because it helps prevent the total extinction of all organisms within a species, because if they did not have genetic diversity within their genomes, all of them would be susceptible to being killed by the same virus, whereas now, since in nature they naturally have genetic diversity, some of them might survive the virus or pathogen and reproduce, thus leading to a more stable ecosystem. Next, let's talk about what are GM crops. GM crops, as defined by the World Health Organization, are crops that have been genetically altered in a way that wouldn't happen naturally in the environment. These genes are inserted into the crops to genetically alter them to give them pesticide resistance and resistance to heat and cold. After they insert these genes into the seeds, they mass produce these seeds on a huge scale, and so all the seeds end up being the same as their specific counterparts. This is important because when we look at facts from the Non-GMO Project, we now know that 90% of canola, 88% of corn, 90% of cotton, and 94% of soy could all be wiped out by 4 pathogens because they are all genetically the same. Next, let's talk about how GM crops are affecting biodiversity on a massive scale. Let's look back to 2004 in a study done by Harvard where they looked at a GM grass farm. This GM grass farm, every gray dot that came from it was a seed that they found, and every red dot was a seed that actually reproduced. These reproducing seeds are the main problem because they reproduce with the grass around it, and then that grass reproduces and spreads, and then that grass reproduces and spreads, and so forth. This ends up creating the whole area around the GM grass farm genetically the same, meaning that it is all susceptible to the same kind of pathogen or virus. This is happening across the U.S. with our crops. Say, for example, you look at the corn in the U.S. The corn is spreading outside of its farm because no one is watching the seeds that it is spreading, and then it's reproducing with the wild version and leaving the whole area for miles and miles around the farm susceptible to the same risks. That's why we believe that GM crops affecting biodiversity is such a big problem. If a pathogen was to get out that would actually destroy these plants' genome, it would leave mass economic destruction in its wake. Everything from the farmers who are actually farming the soil to the people who are buying the food.
With GMOs causing unwanted and unforeseeable environmental problems, we need to be able to test their effects in a closed ecological environment. One way that we can do this is to test new GMOs inside of the Biosphere 2. The official website for the Biosphere 2 states, the University of Arizona Biosphere 2 consists of a unique, large-scale experimental apparatus housing 7 model ecosystems, which means that the Biosphere 2 is already stocked with plants and ecosystems that we can test GMOs in. The reason that we want to use the Biosphere 2 specifically is that it is one of the best facilities for testing the most amount of variables because we will have complete control over climate, temperature, all those other variables. The mission of the Biosphere 2 is to advance our understanding of the natural and man-made environments through unique experimentation, so says the official website for the Biosphere 2, which means that they would likely allow the tests to occur. The pros of using the Biosphere 2 are that we will have complete control over it. Climate and temperature, we can change all of those variables for the most conclusive results. The second pro of using the Biosphere 2 is that there will be no environmental damage because it is fully enclosed. A third pro is that it is ready to start pretty much right now. The problems, however, are that like other GMO tests, it will take time to see results. A second con of using the Biosphere 2 is that the GMOs may cause enough damage to the Biosphere 2 that it needs to be reset, which would slow research down even more and cost a lot of money. A third con is that it can't help with GMOs that are already affecting biodiversity. The major limitation with the Biosphere 2 is that as good as the Biosphere 2 is, it is only one facility. So we only have that facility to use, and it will take too long to produce others.
So our second solution is to make GM crops infertile. Making genetically modified crops infertile means bioengineering our GM seeds to not be able to reproduce within themselves or with other species. This will help decrease the taking over of indigenous species ecological niches. Now, why is solution 2 important? As we know, seeds spread very easily and quickly due to sheer number and evolution. Being made dominant and resilient species, our GMCs are more GMCs are surviving and thriving, while the local natural crops are slowly being overtaken by the new and improved competition. Enmer Imborg has a PhD in agricultural engineering and territory studies at the Norwegian Institute of Gene Ecology. They wrote a paper about a study in Europe that tells of the many implications and limitations of GM crops and being so profound in the wild where they are not meant to be. Their research supports that GM crops are potentially dangerous for ecosystems, and they explore the possibility of making GM crops unable to reproduce with other species. The findings of their study show that we know too little about these GM crops and how they interact with the ecosystem around them and biodiversity to know if they're safe enough to use. Stopping the spread of wild GM crops is a feasible and once researched doable thing, and very crucial to maintaining the future economy. Ecologists with the University of Arkansas found a weedy genetically modified crop called the yellow canola growing in the wild far from any designated field or biolab. They decided to request for further research to be done, and they found that it was spreading from China to Japan. They presented these findings at the Ecological Society of America. A major strength of this solution is that it would greatly help to keep the environment stable and biodiversity balanced while keeping the unindigenous species out of the local, already balanced crop populations. And we will still have strong, long-living, disease-resistant crops and unharmed biodiversity at the same time if we implement our second solution. The GMCs already out in the wild are impossible to contain because no matter what we do to make the new ones infertile, we will not be able to recall or pull or even kill off all of the GMCs already out in the wild. Another huge con is that it would be very hard to make batch after batch after batch of new GM crops every time one is farmed or dies. And we have created these GMOs to be resilient for a reason, to provide more food and more food and for more people, all the while taking up less resources and less land. And we could possibly be without a reliable source of sustenance if we implemented this solution. That is our most major con. And in conclusion of solution 2, over time, this solution will work very well to stabilize our biodiversity, but only over time in the affected regions.
In conclusion, our group feels that solution 1, which is to utilize the biosphere 2, is the more effective solution. We came to this conclusion for two main reasons. The first reason is that utilizing closed ecological environments have been proven to work. A great example of this is the BIOS 3 experiment conducted at the Institute of Biophysics in Krasnoyarsk, Russia. And this photo depicts a portion of that experiment. This experiment examined how well life could be sustained on GM foods. The second reason is that using the Biosphere 2 would take significantly less time than producing enough non-fertile, genetically modified crops to balance out the world's biodiversity. Solution 2 may take decades, while Solution 1 will take a mere months to implement and around 6 to 8 years to get accurate results, according to the Mordridge Institute of Research. With this, it will be easier to implement these as the resources are already available. Some of these resources include various mechanical devices that regulate temperature and provide rain, as well as soil bed reactors that include microbes that clean the air. This graph by the Non-GMO Project depicts the use of genetically modified crops from 2000 to 2010. And as Ryan stated earlier, the use of genetically modified soybeans, as represented by this line above, has been very high, hitting 93% in 2010, meaning that 93% of all planted soybeans are genetically modified. Thank you.
Okay, Amar, we're going to start with you. So, give one specific way that your thinking changed as a result of learning about Ryan Smith's paper or thinking.
Well, in Ryan's paper, he discussed the scientific lens, and one that stood out to me was his discussion of the chain reaction that occurred from the use of herbicides and plant repellent. And in his example, he said that when you place these herbicides on plants or plant repellent, that can then not only affect that insect, but also the insects that consume that plant as well as that insect. I had previously never thought that such a chain reaction could occur in such a small confined area.
Okay. Ryan, give one way your thinking changed as a result of learning about Nick's research.
So the main way my thinking changed while thinking about Nick's research, which was the economic lens, was in my paper I mainly focused on scientific, as he mentioned, and I focused on how it affects the environment now. I didn't think about anything outside the environment. I thought of how we genetically modify these plants and then how we plant them and how they grow into the modern world. But looking at Nick's paper where it talks about the economy and how it affects farmers and how it ends up costing less money for the people buying them, it really changed my perspective on how we should kind of come at this problem. We couldn't come at this problem in a way that regulates GMOs because then farmers would be out of jobs and people's food prices may go up if we regulate them. But we had to find a solution that also helped the farmers in a way that wouldn't necessarily just focus on the environment.
Okay. Andy, in the future, what change would you make to how your group worked together and specifically, and how would that improve your presentation?
I think in the future, if we were to do this again, we would spend more time meeting in person. While we didn't meet a lot, it was usually over a call, which was good because it helped us, well, talk to each other and explain what we wanted to do, like increasing the size of our problem so that it would make more sense for each of our solutions. But that wouldn't have helped us as much as if we were to have met in person, because it's easier to explain, and when 4 people are talking on call, it's very difficult to actually hear what's going on. So I would meet in person more to practice and to just discuss, so then we would be able to more specifically help our presentation.
Okay, and finally, Nick, which one of these guys' work had the greatest impact on how you understood the problem that your group identified?
So for me, so Amar's paper was about so the impact that it had on me was, Amar's paper impact had that it was talking about all the regulations of GMOs, and so we didn't exactly want to go that route because that would, like Ryan said, it would take away jobs, and that wouldn't be good for the economy, and that was what my paper was about. So from taking what Amar said about the regulations of GMOs, we came up with a second solution, which was infertility, and so that's basically regulation of GMOs, but it's not taking away jobs. It is taking away the ability of the GMOs, not the ability of the people.